Okay. Hi everyone. Welcome to our another session of RISE-MICCAI Journal Club. And today we have Dr. Yue Li from the University of Pennsylvania, who, uh, who works on developing multimodal imaging biomarkers using a variety of different MRIs. And Dr. Yue Li completed his PhD back in China in computational and applied mathematics. And today he will talk to us about a paper which came out, I think, in last year's MICCAI called Curvature-Guided Coarse-to-Fine Framework for Enhanced Whole Brain Segmentation. Yue, you can take it away.
Thank you so much for the introduction, Pulkit, and thank you for the invitation. Hi, everyone. My name is Yue Li, and I'm currently a postdoc at the University of Pennsylvania. So my current research is about the brain subregion segmentation and imaging biomarker design for neurodegenerative disease and such as the Alzheimer's disease and related ADRD. So today I will introduce this paper with the title of a curvature-guided coarse-to-fine framework for enhanced whole brain segmentation. It was published at the MICCAI last year, and the authors are from the Dalmor Academy and the Hupan Lab in Hangzhou, China. So to begin with, let's talk about the free surfer. If you've worked at the field of neuroimaging, you probably know it. It's an open source tool for analyzing brain MRI. So you can feed a T1 weighted whole brain image into it, and it will give you the segmentation of the gray matter, white matter, and CSF, and a bunch of the subcortical structures. It's used all over different fields, including the neuroscience, neuroimaging, and clinical research on neurodegenerative disease. So in this slide, I just want to give you an idea. I searched the keyword free surfer at the journal named Alzheimer's and Dementia, which is the official journal of the Alzheimer's Association in this week. And during the past 12 months, there are 288 results which mention the free surfer and which use free surfer as a tool. And these are several examples. People use it for all sorts of things in the field of neurodegenerative disease. For example, we can use the free surfer to extract, to measure the tau protein buildup in a specific brain region and finish the quantification of the tau PET. And we can also use the free surfer to look at the hypothalamus volumes and link it to the SNP and ApoE genotype, which are two risk factors for Alzheimer's disease. And furthermore, we can it's not limited to the Alzheimer's disease. It can also focus on some of other neurodegenerative disorders like the PSP, as well as the cerebral small vessel disease. And in the last example, it can be used to extract the biomarker for the perivascular space and find the association between it and the Alzheimer's disease in or the cognitive normal patient in the old adults. So here is what the free surfer does step by step. First, we can start with a T1 weighted MRI, usually in DICOM or NIFTY format. And this pipeline does scar stripping to remove everything that's not brain. So the scar, the dura, and all the non-brain parts are gone in this step. Next, it runs automatic volumetric labeling using an atlas-based Bayesian method. This combines the actual tissue intensity with spatial prior learned from manual label brains. And in this step, not only the gray matter and white matter, but also the amygdala, thalamus, condate, putamen, and brain stems are also segmented. These labels are used to kick off the white matter surface reconstruction. this pipeline didn't show the specific steps, but it includes the white matter segmentation that can be obtained directly from this segmentation step, and using the method of the first marching cubes algorithm, we can create the surface mesh for the white matter, which is a triangle mesh. And this mesh will go through topology correction to make sure that it's anatomically plausible. And finally, it will be refined using smoothing strategy so that we can get the white matter surface mesh. But so far, even if we get this mesh, this mesh's vertices have no correspondence with any atlas. So we will inflate into a sphere shape, and a spherical atlas registration will be performed based on curvature information, like shown in this map. Red represents the sulcus and green represents the gyrus. Then it will create the pure surface by expanding white matter surface outward. And this pure surface will stop at the gray matter and free surface boundary. And this step will keep the topology unchanged and keep the correspondence between the mesh and atlas so that this correspondence can be used in the next step, which is the parcellation. With this correspondence, we can directly map the labels on the atlas to the surface to finish the cortex parcellation like this color map shown. This is a complex and time-consuming pipeline. So besides free surfer, we know in the past 10 years, deep learning models have been widely used in medical imaging segmentation. The authors of this paper divided them into 3 categories, including the patch-based style, the slice-based style, or subvolume-based style. No matter what style we choose, there is one important key features for this deep learning model. That is, they are end-to-end models, which means they take the whole brain image as input and will spit out the segmentation directly. No topology correction and no spherical registration steps, like what we saw just now. So up to now, we can summarize the advantages and disadvantages of these 2 kinds of approaches. For the free surfer, the advantage of it is that it explicitly uses the geometric features like cortical curvature to guide the alignment and parcellation of cortical areas. Actually, this geometric feature is very important, especially in this field, because the brain cortex is a very thin and highly folded structure, and there is almost no intensity difference between adjacent 2 subregions. If we only consider the intensity feature, it is very hard sometimes to distinguish or to segment the different subregions because there is no clear boundary in between. But if we consider more about the geometric feature, it will help us to locate the specific location of that subregion and improve the segmentation and parcellation results. But like what we mentioned just now, the disadvantage is that these pipelines are time-consuming. For example, the surface reconstruction, the topology correction, the spherical mapping, and surface registration. Each of these steps cost a lot of time. So maybe on average, it may cost about 4 hours to process one case. On the other side, for the deep learning-based model, the advantage for them is that they are very fast, especially with the help of GPU, which can help us to process image even within 10 seconds. So it is very applicable in the real-time clinical application. However, the CNN-based model take more intensity features than the geometric features. So sometimes if we specify it into the global whole brain segmentation, the performance in the gray matter of the deep learning model is not as good as free server sometimes. So the author's idea of this paper is to combine these two advantages together. They propose a framework that integrates the cortical geometric feature into the deep learning neural network. Specifically, it can combine the accuracy of the traditional complex pipelines and the efficiency of the deep neural network. Specifically, it contains these three steps, including the coarse anatomical segmentation, curvature computation, and fine cortical parcellation. And we will introduce these three steps one by one later. But if we put these two models together, we can find that these three steps were used to replace some of the elements in the pipeline of free server, and the deep learning-based method is used when necessary to speed it up. Okay, this is the first step, which is the coarse anatomical segmentation step. They start with a deep learning model trained to do the basic tissue segmentation, including the labels of left and right gray matter, the left and right white matter, plus all main subcortical structures. These subcortical structures are things like ventricles, cerebellum, putamen, and so on. 41 in total for them, and the full list can be found in this link. These structures have clear intensity difference with surrounding tissue so that they can be segmented by the deep learning model directly. And this step is pretty straightforward, just one network. So it's fast and easy to train. And the output is a kind of raw segmentation that already covers the entire brain. Then in the next step, this is the curvature computation. This is where they bring the geometric information that deep learning model often ignores before. The basic idea of this step is that the authors claimed that some of the surface-based pipeline can actually be replaced by deep learning-based model. For example, the topology correction, the spherical mapping, and surface registration. They find that these different steps can have the literature support that they can be implicitly embedded into a deep learning network training process, so that we don't have to design the explicit step for these steps. Instead, we can only keep those essential steps for curvature extraction to to make it in a very fast speed. Specifically, they would use the anatomical segmentation as input and strip away the cerebellum and brainstem and treat subcortical regions as non-gray matter. And then we can extract two surfaces. One is white matter surface and another is pure surface. So in this example here, what we show is just the white surface. And this surface is constructed using the mesh by the method named first marching cubes algorithm. And after this surface are smoothed, and then the mean curvature can be calculated at each vertex. The curvature tells us about the folding patterns of the cortex, which are critical for accurate parcellation. However, this curvature information is only saved inside of each vertex. But what the author want to do is to map them into the volume space so that it can provide the volume segmentation in the next step for the final fine parcellation step. So in the third step, this is the fine cortical parcellation. They take the original T1 weighted image and the curvature feature map as input so that to form a two-channel input and feed them into another deep learning model. This model focus on the parcellation, the gray matter into detailed cortical regions, using both intensity and curvature to make more accurate boundaries. Once the fine parcellation is done, they merge it with the white matter and subcortical segmentation from the step 1. The result is a full whole brain segmentation that keeps the geometric accuracy of the surface-based method, but runs much faster, maybe in minutes instead of hours. So this is the experiment data set. The others tested their method on the public Mind-Boggle 101 data set, which has 101 T1 weighted brain MRIs with very detailed manual labels, including 31 cortex labels per hemisphere and 2 white matter labels, including left and right white matter, as well as 41 subcortical structures. And they divide this data set into 30 cases test set and 70 case training set, which was used to train the model with 5 cross validation. But so far, there is a question that we haven't answered, which is that we haven't specified the specific deep learning model for these 2 segmentation steps. So what the other choose were these 4 state-of-the-art deep learning 3D model, including 3D UNet, UNet, the UNatural, and Swing UNatural. And they tested this pipeline with this model, compared them with their plain version, which is showing the result as this table. So they compared the dice score, the average dice score of the subcortical structure, the white matter, and the all gray matter subregions. And for each row in this table, the first name represents the bankbone name of the segmentation method. The pant one represents the plain model without any 2-step like coarse-to-fine scheme and without any curvature feature get involved. And the pant 2 with curvature feature means they take the 2-step coarse-to-fine step into the consideration, but not consider the curvature. And the final one is the proposed model, which not only consider the 2-step scheme, but also consider the curvature features. So as we can see, no matter for which bankbone, after improve them in with the 2-step segmentation, the dice score improved when it compared to the plain model. And if the curvature feature is get involved, it can be this dice score can be further improved. And because for subcortical structure and white matter, they were segmented from the initial coarse segmentation, and there is nothing to do with the curvature with them, so that no matter with or without curvature, they should have the same dice score. So they only show it in one row. And here, this slide shows the quantitative segmentation results from the test set, comparing the proposed model with the model without the curvature-guided method. One common issue we can see is that in the baseline model, like shown in this example, some of the CSF between cortical folds is often mislabeled as gray matter, leading to unrealistic topology. And this happens because these models struggle to capture the fine geometric details of the cortex, which are critical for identifying the accurate boundary between gray matter regions. And lastly, they also compare this proposed model with 2 state-of-the-art whole brain segmentation methods. One of them is FreeSurfer apparently, and another one is UNest. This paper was published in 2023, and in this paper, the UNest outperformed UNet, outperformed Informer, and unnatural, swing unnatural in the specific task of the whole brain segmentation. So the authors used it as a deep learning version baseline here to make the comparison. As we can see, it's not surprised that the free surfer can achieve the best gray matter dice score because it implicitly take the curvature and geometric feature as into consideration. And a lot of correction step may help it to improve the parcellation results. And the proposed method ranks second in the gray matter, but with much faster speed. For free surfer, it consumes about 5 hours, but for the proposed deep learning model, it only needs 10 seconds. But as for the white matter and subcortical structure, the proposed method achieved the best performance. But this is more from the NUNet, which is the best state-of-the-art tissue segmentation in most of the medical imaging segmentation tasks. So finally, the takeaway for this study is that first, splitting the job into coarse segmentation first and then file parcellations works better than only doing it at once. Second, if we also use the give the model the geometric features like the curvature, we can get even better results for gray matter boundary segmentation. And third, one of the nice things is that this proposed this study is not to propose a specific deep learning model, but propose a pipeline so that we can swap in different models for the coarse and file stages so that it can be adaptive to different deep learning model. Finally, because it's fast, it's very fast so that it's much more practical for real-time clinical use. And before we end today's presentation, a special mention is this recent work by Dr. Khandelwal on ex vivo 70 ultra-high resolution brain MRI. We know that the ex vivo data has higher resolution than the in vivo data. So it would be a good tool to extract the structure information of the brain and find the relationship between the structure and pathology in the ex vivo space. And after this, we can mapping these findings from the ex vivo space into the in vivo space so that we can design more accurate and more stable biomarkers for neurodegenerative disease. However, for the ex vivo data, there are many challenges in the analysis. For example, the greater heterogeneity in scanning protocols, the greater texture complexity, the collapsed large ventricles, the profound imaging artifacts, and sometimes even sampling cuts in the ex vivo data. So these problems make the existing in vivo pipeline like FreeSurfer not work on the ex vivo data. So to address this, Dr. Khandelwal developed a pipeline that combines deep learning volumetric segmentation with classical surface-based modeling. This allowed them to generate accurate DKT-based cortical parcellation in native subject space at only 0.3 millimeter cubic resolution. And then these parcellations were used for vertex-wise structural pathology correlation, linking cortical thickness to Alzheimer's disease hallmarks such as amyloid beta and tau. And this is actually the first large-scale surface-based structure pathology study at this resolution. So if you are interested in the ex vivo analysis and the brain surface cortical parcellation, highly recommend the Dr. Khandelwal's papers. And I think that's all for my presentation. Thank you for your attention, and I'm happy to take any questions. Thank you.
Thanks, Yue. I don't see any questions in the Q&A, but I was wondering, have you applied these methods in your own research? And if so, can you share some thoughts of how you can extend this work? Maybe are there any other geometrical features that can be incorporated in the volumetric space apart from curvature?
Yes, I agree that the geometric feature, especially the curvature, can help. Because for now, our segmentation is not based on the boundary, like I mentioned just now. Different subregions in the brain cortex is defined by the manual protocol instead of the clear boundary in between. So some of this protocol rely on the curvature, or sometimes it rely on the shape, on the geometric features. But for now, if we only use the deep learning based model, like what I did, what I am using all the time, just feed all the image into the deep learning model directly and train it and ask it to output the labels directly. It ignores these geometric features, especially for the curvature of the cortex. So sometimes the mis-segmentation or the over-segmentation is not because of the model is not perfect, but because the feature is not enough for it to extract them. So, for now, I think using such a curvature-based method, we can use it to increase the dice score when it comes to some of the tiny but no intensity difference subregions, especially for those with highly folded and sometimes in the low resolution image. But I haven't tried it in my own research, but maybe I will do it.
I don't see any more questions. So, well, I see a raised hand. Uh, if you want to type your question, Dheeraj, there's
Yeah, I saw one question.
oh yeah, there's a
The
question.
question was, I was curious if different atlas, for example, AAL3 with around 167 labels for subcortical regions, will be the time complexity be the same for the deep learning model? Also, can we say that surface-based geometric feature and volumes to the volumetric features? I'm not very familiar with the atlas in the whole brain segmentation, but I believe if since the DKT is only one of the atlas, and it was developed from another atlas named DK. So I think as long as the vertex registration is accurate, no matter what atlas input, we can finish the parcellation. As for the geometric feature, if we are using the traditional method like spherical parcellation, spherical registration, they were already used inside of this pipeline, like what we see in this here. So I think it indeed adds some values to the volumetric features, but I don't know how much value it can add for the final volume-based segmentation, because for this segmentation, this parcellation is finished in the mesh space instead of the volume space. But this paper that we presented today, they tested that volume feature after ended it into the volume space. The geometric feature ended into the volumetric space. It can help the deep learning model to improve its accuracy. I'm not sure if my understanding is correct, and Phuket is the expert. You can add anything that not so accurate from my answer.
Yeah, yeah, I wasn't aware of this AL3. I'm just looking it up. Uh, yeah, I'm not too sure. I think if you're trying to train a deep learning model with these label, I think the time complexity should not be affected much. But I'm not sure how much adding curvature-based features would help in these small subcortical regions. I think a combination of some kind of surface and some kind of volumetric image together as an input uh, might be more beneficial as well. I'm not sure. Yeah, that's a good question. Oh,
And another question is, which backbone is most efficient when it comes to segmentation related with brain imaging? If the In my understanding, if the backbone is for the deep learning model, I believe NUNet still is still the best one. If we look at the if we look at the comparison between these four different backbone, NUNet is still very accurate, much accurate than others. But as for the efficiency, I'm not sure how much time it will consumes for an unit. But as for my own subject, it can finish one segmentation within 2 or 3 seconds. So I believe the efficiency. We don't need to worry about the efficiency of that.
yeah. I also think NN unit is probably the best backbone when it comes to deep learning segmentations. And the authors of NN Unit have a nice series of papers where they benchmark their papers with respect to recent transformer based architectures as well. Those are good questions. Well, I think if there aren't any other questions, then we should thank our speaker, Yue,
Thank you, everyone.
presenting this paper, and there were nice questions.
Yeah.
Actually, there's one more follow up question. Yue, if you want to read it.
Yeah. One follow-up question. From your experience, have you observed any geometric feature help understanding difference between control and pathology group? I was thinking about application your model for application to different brain regions, difference across groups. So this, in terms of the downstream task, to use the segmentation to use the segmentation in distinguishing between different patient group like control group and disease group, or maybe the mild impairment group with the dimension group. So this is not my paper, so I'm not sure how it will work, but at least for the similar work from the Phuket paper, we can find the significant difference using the extracted thickness from this pipeline when to distinguish different group with different disease pathology. So I believe if the segmentation is accurate, we can find the significant difference between different groups. And if the image resolution is much higher, this thickness or this biomarker should be more accurate. So yeah, so I believe as long as we can get the accurate, highly accurate segmentation, we can find the difference between these groups.
Yeah, I think for downstream tasks, definitely geometric features are used all the time to understand the differences between pathology and controls. But yeah, if we want to include those features within the segmentation or parcellation model, yeah, that's a nice suggestion to see how if that might be helpful. Because if there are diseased brains which have smaller areas, then the geometric properties would be different, and that could help inform the network how to segment same structures but which have severe atrophy than structures which are healthy. That's a good point. Yeah. Uh, yeah, those are great questions. Uh,
Yeah, thank you for the question.
Thanks, Yue, for presenting this paper. I think learned some nice questions and there was some good discussion.
Yeah, thank you. Thank you for the invitation and thank you for your,
Thanks everyone for attending as well. Uh, have a nice weekend.
yeah. Have a
Bye.
nice weekend, everyone.